Welcome back to the Deep Dive. Today we are taking on one of the most persistent and, frankly, elusive questions in human evolutionary biology.
It's a big one.
It is. We're stacking it up against some incredible scientific rigor to try and finally get a definitive answer. We're talking about chemical signals of fertility.
Right. Scent.
Exactly. I mean, in the vast majority of the animal kingdom, and especially among primates, our closest cousins, females, broadcast their fertile window loud and clear.
Often through scent, yeah. Specifically from their private regions. It's essential reproductive information.
It is. And since we share that deep evolutionary history, the question it persists. Do modern humans still possess these leaky cues? Okay, let's unpack this.
And that shared ancestry is exactly why researchers keep looking. But, the track record in humans has been extremely mixed.
To say the least.
For decades, you've had these small studies suggesting women might unconsciously advertise fertility through subtle changes, maybe a shift in facial attractiveness or a higher-pitched voice.
Right, I've seen those. Even wearing more revealing clothing.
Exactly. But every time a large, high-powered study comes along to try and replicate those findings, they tend to just disappear.
The effect vanishes.
It vanishes. And that brings us to the study we're diving into today. The mission here was to test this whole leaky cue hypothesis, the idea that women unintentionally emit physical fertility cues.
But with a new level of thoroughness.
Unprecedented thoroughness, focusing specifically on scent from the vulvar region. And they didn't just, look at the chemicals. They also did these really robust perceptual tests.
So the human element.
The human element. And for you listening, here is the major takeaway up front. Despite all that rigor, the short answer is no. Chemical cues of fertility appear to be largely lost. Now we just need to understand why this study was the one that finally gave us that definitive non-finding.
Okay, so before we get to the results, I think it's important to appreciate the evolutionary stakes here.
Absolutely.
Across almost every group of primates, from tiny lemurs and baboons to great apes like chimps, body odor from the genital area is a reliable form of communication.
It's a billboard, basically.
It is. Males perceive it, they respond to it, and it informs their entire reproductive strategy.
And yet, when human researchers try to locate this signal, they have traditionally looked almost everywhere else. Mostly, they've focused on the armpits.
Which has given us really inconclusive results.
Totally inconclusive. But the vulvar region, it's a perfect candidate for a chemical signal. It's packed with exocrine glands. You've got sweat glands, sebaceous glands. And we know that when estrogen rises before ovulation, the vaginal pH drops. It becomes more acidic.
So physiologically, a signal should be generated there.
It should be.
I think the experimental rigor of this study is its defining feature. They clearly saw the flaws in all that prior research.
They did. They recruited 28 naturally cycling women, all non-smokers, all off hormonal birth control. But what really jumped out at me in the source material was the diet restriction.
Oh yeah, that was intense.
It's extreme, but it's so necessary if you want clean data. To eliminate all these outside factors that could muddy the odor profile, every donor had to adhere to a strict vegetarian or vegan diet.
For at least a month beforehand.
For a month. We know for a fact that meat consumption alters body odor quality, so they controlled for it aggressively.
Wait, so they went to all that effort, the diet, no hormones, just to isolate this pure, essential body scent.
Yeah.
That level of control it's massive.
And they didn't just rely on guessing when ovulation happened, right?
Absolutely not. That's a huge pitfall of the older studies, just relying on calendar dates. Here, samples were collected across up to 10 days, specifically covering the fertile window, but they used sensitive urine LH tests and salivary hormone analysis.
So double confirmation.
Exactly. To confirm the exact day of ovulation and to measure the key hormones, estradiol and progesterone. This hormonal confirmation means they knew precisely which samples came from the most fertile moments.
Okay, so let's move to the lab results. They analyzed, 146 samples? 146 samples, yeah, using gas chromatography mass spectrometry.
For anyone unfamiliar, you can think of GCMS as a kind of high-tech chemical sniffer. It separates a scent sample into all its tiny individual components.
And lets you identify exactly which volatile compounds are present. They looked at over 100 different compounds they thought were likely to be human-derived. And what's fascinating is how they tested the data. They basically asked two different questions of the chemical profile.
Okay.
First, is there a subtle shift that could be tracked over days or weeks? This is the continuous monitoring scenario, useful information for a long-term partner.
I see. And the second?
Second, is there a sudden, unique spike in certain chemicals right around ovulation? That's the immediate assessment. A kind of chemical advertisement for a first encounter.
So, two different ways the signal could work. What did the continuous monitoring, the tracking over the cycle, what did that reveal?
It was a clear chemical non-finding. There was no significant association at all between the overall similarity of the women's chemical profiles and any of the fertility predictors.
So in plain English,
In plain terms, the chemical signature of a woman's scent just didn't change predictably enough for someone or something to chemically track her fertility across her cycle.
wow. Okay, so that's a bust. What about the immediate spike? Was there any evidence of a single powerful chemical advertisement?
Not robust evidence, no. It was a bit tricky. While certain statistical models, specifically when conception risk was tested as the sole factor, they tentatively suggested that 7 substances might have fluctuated with fertility risk.
Okay, so a maybe.
But very tentative maybe, because that connection, it vanished when the researchers ran their robustness analyses. They excluded any samples that might have been slightly contaminated or collected under less than perfect conditions, and the effect just disappeared.
The chemical link just wasn't strong enough to hold up.
Not consistently, no.
So even though they identified things like acetic acid or a urea-related compound showing a slight trend, that trend basically dissolved under intense scrutiny. It's hard to build an evolutionary argument around a chemical cue that can't survive basic statistical filtering.
You can't. And what's even more compelling is that when they looked at those tentative compounds, they did not strongly overlap with the established chemical fertility cues you see in other apes and primates.
Ah, so it's a different chemical language anyway.
It seems to be, which is highly suggestive that whatever signaling capacity we once had, our chemistry has just, it's fundamentally changed.
Okay, so the lab couldn't reliably track fertility chemically. Now we move to the human element. Could the man on the street, the intended receiver of this signal, do any better?
The big perceptual test.
Yes. 139 heterosexual men came in as raters. They were smelling 274 samples. These were cotton pads worn overnight by the women. In a simulation of a first encounter, the men rated each sample on an 11-point scale for attractiveness, pleasantness, and intensity. And here's where it gets really interesting. With all those controls, all that immense effort to capture a clean sample, did the men detect the fertile window?
They did not.
Not at all.
The men's ratings of the women's vulvar sense did not significantly vary with any of the female fertility predictors.
No way.
It didn't matter if the woman was at peak conception risk, far from ovulation, or had high levels of key hormones. The men rated the attractiveness, the pleasantness, the intensity, all pretty much the same.
So they just smelled average.
They just smelled average.
Yeah.
This is a definitive null result, and it was confirmed through really sophisticated statistical models designed specifically for analyzing this kind of ranking data.
And this finding, it directly contradicts all those small earlier studies?
Yes.
The ones that fueled the myth that men find periovulatory odors more pleasant?
It completely contradicts them.
It throws a real wrench into the evolutionary psychology canon, doesn't it? It's just amazing to think that after all that effort to isolate a pure fertility odor, the men just rated everything as a fine.
It does, and it aligns perfectly with the current scientific consensus that's building around this topic. We have accumulating evidence that this signal is simply lost in humans.
So it's not just this one study.
No, not at all. For instance, a separate study that used armpit odor from these very same women also found no evidence for fertility-related shifts. We also know from studies involving committed couples that men in established romantic relationships, they don't seem to detect their partner's ovulatory cues either.
Fascinating. So if we connect this to the bigger picture, the overall data suggests a very low retention of these chemical cues in modern women.
It does. And the critical question, which I think is often more interesting than the null finding itself, is why? Why would humans lose such a fundamental reproductive signal that our other primate cousins still rely on?
Right, the why. The first reason seems to relate to how we changed our physical habits and our culture.
Exactly. I mean, start with locomotion. Other primates walk on 4 legs, which naturally puts that chemical signaling region right near the noses of their peers.
Easy to detect.
Very easy. But the human transition to bipedalism, to walking upright, it physically moved the source of the odor away from the noses of our species.
And then we put clothes on.
And then we put clothes on, add in cultural factors like clothing and hygiene practices, and you have physically covered and culturally suppressed the signal, just reducing its sociochemical relevance.
So we literally covered up the signal, and it stopped mattering as much.
Yeah.
But the internal changes are just as significant, aren't they?
They are. And this is where diet and the microbiome come into play. Compared to non-human primates, humans developed a vastly different vaginal chemistry.
Because of farming.
Largely, yes.
Yeah.
Our agricultural revolution, with its higher starch intake, led to higher levels of glycogen and a dominance of lactobacilli bacteria in the human vagina. And these bacteria drive the environment to be highly acidic. They produce a lot of lactic acid.
Which would change the smell.
It fundamentally altered the composition of vulvar volatiles. It pushed them away from the chemical cues we see in other mammals. In short, our diet changed our biology, which changed our scent profile, and that diverged us chemically from our closest relatives.
And the third reason is maybe the most shocking to me, because it deals with the very mechanism of fertility itself.
It is surprising. I mean, despite the fact that ovarian hormones, estradiol, and progesterone are the central mediators of conception probability,
They're running the whole show.
they're running the show. But this study found that they did not significantly influence the vulvar volatile profiles. The key reproductive hormones are simply not consistently driving changes in this odor signal.
So if the signal doesn't consistently respond to the drivers of fertility, it's not a functional cue.
It's not a
Not it.
functional cue. It's broken.
So here's the clear takeaway for you. This highly controlled, two-pronged study suggests that the strong, clear chemical fertility cues we see in our closest relatives, they appear to be largely lost. Or at least, they're simply too inconsistent to be reliably tracked or perceived by men in a first encounter setting.
The robust conclusion is that we just do not support the leaky cue hypothesis when it comes to vulvar scent. And now we need to continue this large-scale comparative work with non-human primates to really trace the evolutionary history of these vanishing signals.
And that leads us to our final provocative thought for you to consider.
Yeah.
This study was conducted in a controlled lab setting, simulating a first encounter where men were just sniffing a cotton pad.
A very artificial context.
Very. And the researchers themselves suggest that perhaps perceiving vulvar scent requires an intimate, potentially sexual context for an effective response. So could the cues be functional, but only in the moment of intimacy where secretions change drastically during arousal, rather than during a passive assessment at zero acquaintance? We'll leave you to mull that over.